This video will be an introduction to our next lab project on gene annotation. So this project is actually a collaboration with a group of faculty and students at other institutions. Our organization is called the Genomics Education Partnership, or GEP. The goal of this organization is to get undergrads involved in authentic genomics and bioinformatics research experiences in the classroom. And the idea of this is that we have this overarching science question, this comparative genomics research question that we address by having lots of different students and different institutions each contribute in a very small way. So this is an overview schematic of our workflow for the GEP, and I just want to highlight the final goal is scientific publications in comparative genomics. So we start off with these publicly available genomes, which are really considered draft genomes because no one has carefully identified where the genes lie on these genomes. A key early part of this process is to actually compile that genetic sequence into Genome Browsers. The UCSC Genome Browser is what we'll be using. And our staff are the ones that do that. And our staff are located at Washington University in St. Louis and also at University of Alabama. And then based on the research question that we are addressing, and we have several different ones, we have separate projects that the GEP faculty claim for their students to complete these annotation projects. And so at the individual institutions, students finish these annotation projects and then they submit the information on where those genes lie back to the GEP. They get checked and eventually we use them for our scientific research. The project that we will be involved in this semester is called the Pathways Project, and we will be using this genus of Drosophila to address this question. We will be looking at the conservation of genes within a particular signaling pathway called the insulin signaling pathway, and we will be investigating how the evolutionary conservation of these genes is affected by the position of each protein in a network of proteins that they interact with. So the process of gene annotation can really be broken down into something this simple. There's a DNA sequence here, and we're going to use evidence and computational assistance to locate where a gene lies on that raw DNA sequence. So the types of things that we will be looking for are the individual exons of a protein coding region of a gene. We will be looking for the start codon at the beginning and the stop codon at the end of a gene. We will be looking for the splice sites to locate where is the beginning of an intron and the end of an intron. And I just want to highlight that there are other parts of a gene that we've talked about, such as the untranslated regions shown in teal here. And we will not be trying to locate the untranslated regions in this particular project. So we will really only be looking for where the protein coding sequence lies within a gene. There are a lot of tools and programs that have been written to try to aid in this process of locating where genes are on raw DNA sequence. And I just want to highlight that at this point, human curated analysis is actually more accurate than any of these computational approaches. And so even someone who's relatively not an expert at this, humans are better at synthesizing the various pieces of evidence to come up with the best possible model of where that particular gene lies on the DNA. And so our goal is to really collect and analyze and synthesize all of that evidence to create the best possible prediction of where that particular gene lies. Also, I'll just mention that the approach that we use is really optimized for the situation in which we have a very well understood and well annotated reference species, which is a related species that is fairly close evolutionarily. So in the process of locating where genes lie on chromosomes, it's important to remember what the structure of genes is. So we start transcription at the beginning of the first exon, and we stop transcription at the end of the last exon. After we create that pre-messenger RNA, or the primary transcript, the introns are removed and we end up with the final messenger RNA, which includes both the protein coding sequence and also the UTRs, or the untranslated regions. And so some of the terms that we will use are the entire part that is transcribed, we would call the gene span. And the parts that end up in the final RNA would be called the exons. The untranslated regions, or the UTRs, are on either end of those RNAs, and the protein coding regions, we have actually a special name for those, which may not be familiar. It's called the CDS, or coding exons. Just want to highlight that since we will only be looking at which regions are protein coding sequences, the goal of this project is to locate the start codon, to locate the stop codon, and to locate where all of the introns lie. We'll use three kinds of evidence to annotate genes. So the first very, very important evidence is conservation with our reference species. The second kind of evidence is what ends up transcribed into RNA, or the RNAseq evidence. And the final one is computational evidence. And I'll be going into all three of these in a bit more detail. Throughout the entire process of annotation, we will pay careful attention to following basic biology rules. So first we'll talk about conservation. Conservation is really the similarity with our reference species, which is Drosophila melanogaster. The main type of conservation people usually think about is the conservation of protein sequences, and we certainly expect that to be true. So as species diverge from each other, they get more and more different over time. However, the parts that are important, specifically protein coding sequences, tend to stay more similar because they are functionally important. In addition to the conservation of protein sequences, we also are going to expect that we have other kinds of conservation as well, specifically conservation of the structure of the gene. So a gene in melanogaster, if it has 4 exons, that we would expect in a closely related species that species would also have 4 exons in that particular gene. We also expect that if there are alternatively spliced variants, so different mixing and matching of different exons in alternative isoforms, that those will also be conserved between closely related species. We also expect the overall size of exons to be maintained. Finally, I'll just mention there's an additional type of conservation that we will not be exploring in this video. This is conservation of the chromosome neighborhood. And so the same gene in a related species, we expect that the genes around that particular gene will also be the same, and their orientation will also be conserved. So most of our consideration of the evidence for gene annotation will be done within a genome browser. And a genome browser is shown here, and this is simply displaying a region of DNA sequence from a species. It's actually too zoomed out to see the base pairs, but all of the evidence that is associated with that particular region of the genome is displayed in these different tracks that are found underneath the sequence itself. So there are two ways that we will be using conservation within the genome browser. The first thing is that conservation is actually going to direct us to exactly where each coding exon is located within this genome. We will be directed to that location by blast searches with each exon. Then once we get to the region of the browser, there's another conservation track that will be used, and this is this SPAR ALN alignment to D-melanogaster proteins. So this is not telling you that this for sure is that particular gene. What it's telling you is that when that particular genome sequence was blasted against all of the melanogaster proteins, there was a region here that was very similar to a Drosophila melanogaster gene called ILP8. The next type of evidence is RNA-seq, and this is really looking at what ends up in the RNA. As you all know, the entire gene span is initially transcribed, including exons and introns, but introns are removed immediately from that RNA to produce the final processed messenger RNA. Introns, because they are removed immediately and they are not protected from degradation, they immediately are degraded. And so what really shows up in RNA-seq data is what is in the final messenger RNA. There are a couple of different tracks on the Genome Browser that tell us about RNA-seq data. First of all, there is this top one called the NCBI RefSeq genes. This is actually a curated version of the RNA reads that are from this particular species. If several different RNAs are present for a particular gene, then there may be several different RefSeq predicted genes in this blue track up at the top. The second one are these little peaks, and the height of those peaks represent how many times a particular sequence was found in the messenger RNA. In this particular example, we had two different samples of RNAs from adult females and adult males. And when we see these high peaks, it's a nice indication that probably a gene is found in this region. Finally, we have splice junctions, and these are just a particular subset of the RNA-seq reads, and they specifically include only the ones that map to two different regions in the genome. So for example, this brown one down here actually had a little part that was similar to this region right over here, and it had another part that was similar to this region over here. The line was actually not found in the RNA, and therefore it's very strong evidence that in at least several RNAs, this was a region that was spliced out of the messenger RNA and likely is an intron. Finally, computational evidence is our weakest form of evidence. This includes information from programs that have been written to predict the location of genes in newly sequenced species. These are called ab initio gene predictor programs, and these should be really used only when other evidence is lacking or as backup evidence. There are three different tracks that are displayed here. Gamoma, Gene ID, and Augustus are all different gene prediction programs. And you can see that these three do not agree with each other where that particular gene may be found in this region of the genome. The last thing I'd like to discuss in this video is which are the very important basic biology rules that we need to pay attention to. So the first thing is that introns are defined by splice donor and splice acceptor sites. And so when we look at these particular sequences shown with these orange arrows in this 3 exon gene, we would always look for a particular sequence at the start of an intron. This is a splice donor, and the first two bases are always a G followed by a T. At the end of introns, the last two bases of an intron are always an A followed by a G. These sequences must be there for the cell to realize that that is an intron and to effectively cut it out of the messenger RNA. So we'll look for those. One thing I want to mention is that very occasionally in Drosophila species, you can find a G followed by a C as the splice donor. But most of the time, like 99% of the time, it's actually a G followed by a T. Importantly, not all GTs and not all AGs are splice sites. So there are additional sequences that are preferred sequences for the spliceosome to recognize that position as the start or the end of an intron. But we will really only focus on those 100% required sequences. The second major biology rule I'd like to highlight that we are going to have to pay careful attention to is the open reading frame. So for eukaryotic genes, we have these individual coding exons or CDSs, and there is one particular reading frame that is the correct reading frame. I just want to highlight that different coding exons might in fact use different reading frames, but we know which reading frame for each individual CDS is the correct reading frame based on two pieces of evidence. First of all, there's an absence of stop codons in the correct reading frame. And second is that the sequence itself is a conserved sequence. It is encoding a protein that is found in another species. So each CDS has its own particular reading frame that must be used. And when those individual exons are spliced together, the correct reading frame must be maintained. The way that we are going to track this is by paying careful attention to something that we call the phase of an exon-intron boundary. So the phase of each exon-intron boundary is the number of bases that are in the exon that are not part of a full codon. We're going to keep track of these extra bases and make sure that when two exons are spliced together, that any extra bases are brought together to make a full codon of 3 bases. So an entire codon would be 3 bases, and so the number of extra bases could be either you could have phase 0, you could have phase 1, or phase 2. I want to illustrate this by looking at the possible ways that a particular codon might be split between two different exons. So here's an RNA with the codons in the correct reading frame indicated. And this particular codon, pay attention to this one because right in the middle here is where we're going to split between two exons. So one possible way that this might be encoded in the genome is shown in this first one. So here we have the intron in orange. We begin with a GT, we end with an AG. And this entire thing would be cut out in the splicing process. So occasionally we'll have situations where we split the coding sequence exactly so that there's no extra bases that aren't part of a codon at each exon-intron boundary. This would be called phase 0. So the last 3 bases of that particular exon are AGA, and so there's no extra bases, and so we just call it 0. And so if we have a phase 0 splice donor, it must attach to a splice acceptor, which is also phase 0, because we don't want any extra bases to put the next coding exon out of the correct reading frame. Another possible situation we might encounter is that that codon is actually split between two exons. So you can see that GAT is split, and one of those bases, the G, is in the first exon, the AT is in the second exon. This one, we're going to have to keep track of these extra bases, and there's one base here that's not part of the codon, so we call it a phase 1, and there's 2 extra bases here, so it's a phase 2, and we have to have a phase 1 hooking up with a phase 2 to make a codon of 3 bases. The final possibility is that you have 2 extra bases at the first exon and the 1 extra base at the second exon. This would be phase 2 and phase 1. And again, that needs to add up to 3 so that we can create a codon when those 2 exons get spliced together. Now, the tricky thing when we look at the genome browser is that the phase of an exon-intron boundary is dependent upon which reading frame you are using for that particular exon. So when we look at the bases in the genome browser, when we zoom in far enough, you can actually see these individual bases, and the 3 different reading frames are shown below them. This would be reading frame 1, 2, and 3. If this particular exon was being translated in reading frame 1, we would pay attention to this top line of protein sequence. In this dark red box is the splice donor site. So the GT, that G is the very first base of the intron. This will be removed during splicing. What we want to know now is the phase, how many extra bases are present in the exon that are not part of a codon. And so if we are in reading frame 1, we would notice that the very last full codon within that exon is that W encoded by the TGG. And there are no extra bases that will be in the exon after that intron is spliced out. So therefore the phase would be 0. However, for this exon, if the correct reading frame was reading frame 2, we would look at the middle line here for the correct encoded amino acids. If we look at where the very last full codon is, you would see that it's that R, which is encoded by CGT. That's the last full codon in that exon. And there's actually 2 extra bases after that. There's 2 Gs that are going to be part of the exon, but are not part of a full codon. And so therefore, the phase of this one, if it was in reading frame 2, would be phase 2. Finally, if we were looking at reading frame 3, the last full codon would be the V that is encoded by GTG, and that leaves 1 additional base, that G, which is not part of a full codon, but is part of the exon. That 1 G means that the phase would be 1.